Hello and welcome to Asexual Reproduction, Day 3. So today we will be talking about mitosis more in depth. So today's aim is how does mitosis form identical daughter cells? And so mitosis, what is it? We'll find that out today. And remember that these identical daughter cells have exactly the same genetic information. And piggybacking off that, let's start with the do now. Why must every cell produced by asexual reproduction receive a complete copy of DNA? So your DNA is the blueprint or the instructions for life. So if you've ever tried to put together like a Lego set or a piece of furniture that has instructions, probably not tried to build a house from a blueprint. I know I haven't, but if you have, that's pretty cool too. You know that if you don't have the complete set of instructions, if you do not have the complete set of instructions, you won't be able to build your Lego set. You won't be able to finish your piece of furniture. So in order to produce another organism asexually, a complete copy of DNA must be duplicated and passed on. Especially because with asexual reproduction, there's only going to be one parent. There's no other contributing DNA. So in order to produce another organism, needs DNA, and asexual reproduction is only one parent. So how exactly does that replication happen? Well, we'll talk more about that in today's handout and also in Unit 7 when we talk more about DNA replication and genetics. So part A, cell division. Cell division is the orderly separation of one cell into two. Before a cell divides, the genetic information in the DNA of the cells is duplicated or copied exactly. During cell division, one copy of this information is distributed to each cell. And there can be problems with that process, but this is assuming things go according to plan. So during cell division, the DNA is first duplicated within the nucleus, and then the two nuclei split into two genetically identical nuclei, asexual reproduction, and then into two separate cells. As a result of this process, each new cell has all the information it needs to function properly, all of those instructions. One-celled organisms make use of cell division for asexual reproduction. Multicellular organisms mainly use cell division for growth, cell replacement, and for healing from injuries or repair. This process by which a cell's genetic material divides, creating two complete sets of the cell's genetic material, is known as mitosis. And this process is happening inside of us all the time. Mitosis produces two cells that each have a full set of identical genes and chromosomes. And this is where a problem can occur unless a mutation occurs along the way. So And I will just give a caveat to this entire lesson that we are really steamrolling some complicated concepts here and simplifying it for the sake of living environment. So, part B, memory jogger. Recall that DNA replication makes an identical copy of all genetic information in the molecule. And that's a big, long, complicated molecule. The replica carries instructions for the same proteins as the original strand. When the DNA replicates, it is actually turning a single-stranded chromosome into a double-stranded one. The double-stranded chromosome then has a duplicate set of instructions to pass on to each of two new cells. In the image below, observe chromosome duplication resulting from DNA replication. As a result of DNA replication, the chromosomes become double-stranded. So you can see here, this is a chromosome, and this one single structure is a chromosome. And here we're looking at two chromatids. So what happens here is this chromosome is all condensed, it's ready to replicate. And you can see here it has this sort of central focal point called the centromere. So when the chromosome is about to duplicate, that centromere actually is not replicated because it sort of holds the two sister chromatids, the one original and now the copy, in place. And those two sister chromatids contain all of the exact same genetic information. They code for the same proteins, and now they are complete. After they have been replicated, they will go into two different cells that will each have a complete set of genetic information. So we have here this chromosome and then the two sister chromatids, and they're sister chromatids because they're connected by this centromere here, but they will go on to carry out their lives separately into separate cells. So let's look a little bit more at chromosomes and sister chromatids by looking at mitotic division. So during the process of mitotic cell division, the double-stranded chromosomes that are visible during mitosis split into two identical strands and move apart to opposite ends of the cell. Sort of like what we just looked at, right? The double-stranded chromosomes that are visible during mitosis split into two and move apart to opposite ends of the cell. In the image below, observe a chromosome during mitosis. When the cells divide, each double-stranded chromosome separates into two identical strands. The process concludes when the cytoplasm divides, resulting in two smaller but genetically identical cells. So this is an example here of homologous chromosomes. So these are chromosomes that are coding for the same genes, but they are different from each other. They don't have the exact same, they don't code for the exact same proteins. They might identify the same genes, but they don't code for the exact same proteins. When replication happens, these two sister chromatids move on to be in separate cells. And this diagram is representing chromosome replication as it will occur in mitosis. And later, when we look at meiosis, this part of the process becomes a lot more complex. And that is where the unique variety of meiosis really comes into play. But let's keep talking a little bit about mitosis. So here in mitosis, we are looking at the different phases here. So we start with interphase, which is where the cell is at most of the time. This is sort of pre-mitosis, but this is where the DNA is copied. And this is when the cell is growing, developing, and preparing for mitosis. Then we have prophase, which is traditionally the first phase of mitosis. So prophase is when the duplicated chromosomes condense, and you can see that here. Those chromosomes would be visible under a microscope, and we'll take a look at what some of those chromosomes actually look like under a microscope during class, which is pretty fun. But prophase is when the duplicated chromosomes condense and the nuclear envelope dissolves. We would normally have you draw this. So if you're finding this process to be really confusing and you're like, what do these words mean? My word of advice to you is to draw out this diagram. Once I had a student bake cookies and ice them in mitosis. It was really cool. You don't have to do all that, but you can see here all these different parts of the process are illustrated here in this diagram. So we can see the duplicated chromosomes condensing, becoming really visible here. And this is the nuclear envelope, right? So sort of like the cell membrane, but for the nucleus, and it dissolves as the cell is preparing for replication because this is going to become two distinct and separate nuclei. So the first nuclear envelope needs to dissolve so that new ones can be created. So in metaphase here, the chromosomes now line up along the cell's equator down the middle of the cell to make sure that all the DNA has two exact copies that are going to separate, and split into the two different cells. This is where some errors can happen if the splitting is unequal or there is some error in the duplication. This is where you might not get a daughter cell, right? And we'll talk more about that when we get to Unit 7. But so now in metaphase, the condensed chromosomes line up along the middle of the cell. And then my favorite phase of mitosis, I just really like it. It's anaphase, and she's groovy. Anaphase is a girl, and anaphase is when the chromosomes separate. Anaphase isn't actually a girl, just spiritually in my mind she is. And I just think that anaphase looks really beautiful when you observe cells under a microscope. It's always like the anaphase just really jump out at me. And we'll take a look at mitosis in onion cells during class. And you can see the chromosomes are separating. And so this is the border that will become the cell membrane of the other two cells. It just isn't there yet. So in anaphase, the chromosomes are separated and pulled apart to opposite sides of the cell. So that is when those sister chromatids will be separated. They're being pulled apart and dragged to opposite ends of the cell. And there's sort of like an unraveling of the chromosomes that were connected by that centromere being pulled apart, and it looks really beautiful under the microscope. Anaphase, she's cool. And now we have telophase and cytokinesis. that are sort of put into two, but telophase is really right before cytokinesis, right before there are two separate cells. You have telophase where it's clear that they are going to be two separate cells, sort of almost looks like a part of binary fission. That is telophase. And then cytokinesis is the actual cell splitting to form two identical, in this case, skin cells. But telophase and cytokinesis are when the nuclear envelope reforms, and you can see that here. Now it's actually a cell again, or in two distinct cells at that, because the DNA is housed inside of a nucleus again, instead of being sort of out in during the process of division. We have the nuclear envelope reforming and the cytoplasm dividing, splitting into two identical cells. And that is mitosis. So today we talked about the phases of mitosis. We discussed mitotic division and the replication of chromosomes. We discussed how DNA is actually replicated. We defined cell division, and today's aim was, how does mitosis form identical daughter cells? So all right, that was asexual reproduction day 3. I hope you guys have a super fantastic day, and I'll see you all tomorrow on Zoom. Bye, guys.